thank you for the opportunity to give you guys a bit of a glimpse in what I've been working on in proximity pharmacology, and specifically how I've been using ICM in kind of all my analysis, and also the tools specifically. Let me make sure, yeah. But what is proximity pharmacology, you might be wondering, and I think most people are more familiar with the term PROTACS. PROTACS are standing for proteasome targeting chimeras. And proximity pharmacology in general, we can define it as a compound that brings two proteins in close proximity to elicit the response of one protein onto another. And again, for PROTAC, in that case, is the recruitment of an E3 ligase to a target by a compound to induce the ubiquitination and the subsequent degradation of the target. PROTACs is kind of paving the way, and currently there are about 12 PROTACs in clinical trials, including degraders for cancer and autoimmune diseases. And an example of that would be, for example, the estrogen receptor degrader by Venus to target breast cancer. But what do PROTACs have, like what are the advantages when you compare them to more traditional small molecule drugs? Is for PROTACs, you need a lower dose compared to a small drug because it has a different kind of mechanism, namely a catalytic mechanism, than an occupancy driven. So if you can see here, with a small molecule, most of the time it needs to stay bound to a target to have an effect, but once it moves away from the target, it no longer has an effect. Whereas a PROTAC can bind and induce kind of this reaction of ubiquitination and then can be recycled back into to induce another ubiquitination event. Also, we can use E3 ligase expressions to improve tissue selectivity, and this is, of course, very interesting in terms of minimizing off target effects. And as have already been mentioned by many speakers before me, there are a lot of undruggable targets, including transcription factors that have very shallow binding sites that bind DNA that mostly are very hard to drug. But since with PROTACs, you don't necessarily have to bind to the main pocket, but can bind anywhere on a protein, you can kind of inhibit the transcription factor by promoting degradation of a transcription factor. But the field has also kind of moved on beyond PROTACs and have moved into the realm of inducing any post-translational modification with, for example, a FIC, a phosphorylating inducing a chimera that recruits a protein kinase to induce phosphorylation, or the reverse by recruiting a phosphatase to remove the phosphorylation, or the reverse of a PROTAC that recruits a deubiquitinase to remove the ubiquitination of a target. And important of this kind of compounds, and is kind of an important part for next session for most of my talk, is that these compounds consist kind of three parts. We have kind of the chemical handle that's designed to recruit a protein modifying enzyme. And most of the time, this is a starting point. The starting point is a small molecule that we know that binds this enzyme, a small molecule that binds a target protein, and then we're connecting these with a linker region. And this linker is very influential because different lengths can have cause different activities of these PROTACs or ProxPharm compounds, but also the composition of these linkers are very important. So what I kind of wanted to talk about in this realm of proximity pharmacology is kind of like two parts. Within this like three-part compound, you can of course look into more the small molecule development and looking for new opportunities where we can recruit maybe new enzymes to induce other post-translation modification, but also looking at that linker region. How can we optimize that linker and see which ones would potentially be active versus inactive. So I'm going to be talking about discovering new opportunities in protein modifying enzymes, and also about benchmarking PROTAC versus screening protocols, where I included the ICM protocol, and also a bit more on like how we can optimize this PROTAC virtual screening to filter PROTACs. But first I will be talking about the new opportunities. The new opportunities I look for are in the form of non-catalytic pockets in protein modifying enzymes. And you might be wondering why non-catalytic. Is because we want to keep the catalytic site available for the reaction that we want to take place. For example, in a protein kinase, we want to keep the catalytic site available for the protein phosphorylation. So I either define a non-catalytic site by distance or whether it's in a non-catalytic domain. And I aim to kind of build this database with all these pockets and their information on where they're located, whether there's any chemical matter already there, or for example, reactive cysteines for potential warheads. So how did I do this structural analysis? I did this all in ICM by preparing the structures and then running the ICM pocket finder module to identify pockets in these structures. Then I used the first approach. So by looking at non-catalytic domains, I used the Interpro database to kind of map these regions onto the structure, and then kind of marked which pockets were in these domains. The other approach was to look at the distance. So using like databases that contain information about catalytic site residues, and then basically measuring a distance from these catalytic site residues to mark which pockets are very close to the catalytic site. Then based on filters, whether it's marked as a catalytic pocket or as a non-catalytic pocket, and also durability, I'm left over with these pockets that were deemed non-catalytic by approach and also are druggable. And with this structural analysis, I was able to retrieve pockets for which we previously knew they existed and had been used to develop ProxPharm compounds, including a pocket in the C1 domain that's found in 23 kinases that normally binds a 4-bol ester, but that has been incorporated in the creation of a phosphorylating-inducing chimera, but also a pocket in the bromodomain that can be found in acetyl transferases to create a ACE tag to induce the acetylation of the FKBP12 fusion proteins. But more interestingly is that I found novel pockets with chemical matter ready to be incorporated in ProxPharm compounds. For example, I found a allosteric pocket in a kinase domain with already an activator bound there that potentially could be incorporated as a FIC, but also a pocket in a non-catalytic domain, namely the zinc finger domain of the HDAX6 deacetylase, which could be incorporated and would be a first of its kind to be a deacetylase targeting chimera. Overall, I found a lot of sites and I used ICM to kind of also visualize these sites and to check for the accuracy of these sites and whether they would be truly druggable. But overall, I found a lot of pockets in a lot of protein modifying enzymes. And I created this database called ProxyBind, where I stored information on the pocket location, potential candidate ligands, if they were there in the crystal structures, and also kind of a confidence level. I assigned kind of a confidence level to these pockets based on whether there was already ligands bound or fragments bound or peptides bound, whether a homologous protein had something bound in that pocket, or whether the pocket was completely novel and it's unsure if it's potentially druggable, but at least came out druggable with the filters. And I've published this as well in a manuscript. Then the next part of my story is, okay, I've looked at the chemical handle development on like the protein modifying enzymes, but of course, a part is also the optimizing of that linker region between those two compounds. And that's kind of what I started assessing. I wanted to kind of know for myself in the future, which program should I use? And that's where I kind of started benchmarking PROTAC virtual screening protocols. Because as we know that PROTACs are to rationally design PROTACs can be very difficult, because what we know is that PROTACs induce what we call a ternary complex, basically a complex that consists of 3 parts: protein modifying enzyme, target, and the compound in between, and that this complex induces novel protein-protein interactions. And these are hard to predict, since these are not necessarily observed in nature without the PROTAC being there. We also know that different linkers can induce different conformations, and that different linkers can have different activities, but also some linkers, different linkers can induce the same conformation and can have the same activity. So it's hard to see what's kind of the pattern in that. And we know that the PROTAC activity is dependent. So I was kind of like interested to see how tools were handling this problem. Oh, I had animations there. So first, one I use, of course, ICM. ICM has a PROTAC model script that's available in the software itself, where it takes as input the 2 proteins and the PROTAC, and kind of creates this ternary complex, and then performs a Monte Carlo simulation where the linker region is flexible, and you can also opt for the side chains that are surrounding, or like kind of on the closely to this linker region, and the chemical handles to be flexible as well, and then ranks these generated complexes based on the energy of the complexes. And I compared ICM to another software called Mo that has a different approach. It first does a protein-protein docking, then performs a PROTAC conformation search, and then kind of matches these 2 up to see what is viable ternary complexes. And ProTAC has kind of a similar approach, which starts with a protein-protein docking, and then a conformation search within that protein-protein docking complex, and then as well sees which ones are viable. Mo and ProTAC are different to ICM in regard that they rank the PROTACs based on, they cluster their ternary complexes, and they rank the PROTACs based on the biggest cluster that they can find, like the size of the cluster. ICM, the script does not necessarily rank PROTACs, and it's not necessarily a PROTAC virtual screening script, but rather a ternary generation script. So to compare ICM to Mo and ProTAC, I created another analysis that's similar to Mo and ProTAC to evaluate, potentially see if ICM could rank PROTACs effectively. But I also wanted to compare the methods to softwares that do not include any information from the PROTAC. For example, AlphaFold, which is a machine learning program that takes as input a sequence, predicts the 3D structure, and the AlphaFold multiplier predicts also how these proteins interact with each other. And you have HADOC, which is kind of a standard protein-protein docking software. And I looked at 2 questions. I looked at whether these tools can predict crystal structures of the ternary complex accurately, since some of these ternary complexes have been crystallized. And also, can these virtual screening tools dissociate between active and inactive PROTACs? So when I look at the first question, so how well can these tools predict a structure that's closely resembling the crystal structure, I would see that some computational methods, and mostly the methods that include information about the PROTAC, are able to obtain a near-native complex. A near-native complex is kind of defined as having lower than a 10 backbone RMSD, 10 angstrom backbone RMSD, whereas compared to methods that do not include any information about the PROTAC, for example, AlphaFold and HADOC, we're not able to obtain any near-native complexes. And you can see here in ICM performs pretty well in actually finding something that's very closely resembling the ternary complex. But interesting, of course, is where does this ternary complex rank among all of them? And we can see that that is kind of sometimes we are able to really see a complex within the first 100 structures, but not necessarily always the top ranking structure. For example, for Mo, it even sometimes can be found in the 10,000s rather than the top 100. Similar for ICM, most of the time we can find something very similar, sometimes not. Where PROTAC, most of the time, not at all. That was kind of addressing my first question, but my second question was, from a point of view, if I want to use these tools to predict PROTAC activity for potentially which PROTACs do I want to synthesize and start creating. Again, this is part for ICM, is not part of the script. It's something I did myself. And overall, what we can kind of see is predicting activity is a bit unclear. And I looked at this by a fold enrichment, so how many actives are in the selection, the ratio of actives compared to the ratio of actives in the data set. And for some test cases, this ends up with an increased fold enrichment when you look at top 1, top 5, but for some test cases, this is not the case. So that kind of led me to my third part, is like, can we optimize these PROTAC version screenings for them to better filter for active PROTACs versus inactive PROTACs? And I thought of two possible strategies to optimize this PROTAC screening. One by potentially identifying optimal ubiquitination zones within this whole multi-protein E3 colon ring complex, but also maybe learning from conformational difference between active and inactive PROTACs. And for the first one, we kind of hypothesized that the distance could be critical for PROTAC activity, since the E3 ligase is part of this bigger multi-protein complex. So if you look here, this is the E3 ligase VGL, for example, with in green here the PROTAC bound, and in this case a bromodomain 4 binding domain 1 in light blue. It's kind of connected with this ring to the RBX1 protein that's connected to the E2 ligase that's actually responsible for this ubiquitination. And we hypothesized that this distance might be indicative of PROTAC activity, that active PROTACs might induce a closer distance to this E2 ligase versus a inactive PROTAC. So for this, I used a kind of a script that I created myself in ICM, where I compared the number of structures that have a lysine in a certain cutoff, and the number of lysines that could be ubiquitinated in the cutoff. And for both, I kind of see that there's not necessarily a difference between active and inactive PROTACs in either generating a structure that has a lysine in this cutoff, nor the amount of lysines that are within that cutoff. So next I thought, okay, maybe there is a ternary complex conformation that only active PROTACs induce, and which is not accessible, or an inactive PROTAC is not able to induce that conformation. So I did that by comparing cluster centers between active and inactive PROTACs, and maybe see if there are conformations that can only be induced by active PROTACs. And for that, I saw that if I look, for example, here, each dot represents kind of a conformation, a ternary complex conformation between the E3 ligase and target. And on the axis shows the number of actives that have that conformation, can generate that conformation, and an inactive, that there are conformations that are only found among the generated complexes by active PROTACs. And as you can also see here, if the colors represent a certain PROTAC, is that some PROTACs are able to induce many of these conformations. Sometimes a PROTAC can only induce one of them. But then if I look overall over all the test cases that I had, it works sometimes, mostly often. At least I'm overall able to see at least kind of one conformation that's only induced by actives. but it's not always representative of all the actives I have in my data sets. So we need more data, and we need more further evaluation of whether this might be a potential, might be potentially very important for PROTAC design in the future. So that comes kind of to my conclusions, that there are many more opportunities to explore in proximity pharmacology beyond protein degradation if we look at other post-translational modifications that we can influence with these kind of ProxPharm compounds. That PROTAC virtual screening methods are able to predict a neonative ternary complex, and they're quite effective in producing a lot of ternary complexes, but rarely these complexes are a top prediction. But this also begs the question whether the crystallized complex that we observe, is that the only conformation that a PROTAC can induce, or are there multiple conformations? Also, since the crystal structures are kind of a snapshot of the complex, that's of course also another interesting question to keep in mind. We also found a bit of an unclear conclusion for PROTAC virtual screening efficiency, where the geometric distance is not necessarily predictive of PROTAC activity, but there might be something in that active PROTACs only induce a specific conformation that's not observed among inactive PROTACs. And with that, I kind of want to say thank you again and acknowledge the support I get from my supervisor and my committee, but also the group, but also on the side of Molsoft with Eugene, Max, and Andrew, specifically Eugene for answering my questions very rapidly on any problems I run into with Molsoft and with ICM. So with that, I'm ready for any questions there might be.
Thank you very much, Evan. Thank you. Very interesting talk. Any questions?